my lab's work uses patient-derived organoid models of individual tumors. And so what we do is to take primary tumor samples from the clinic, dissociate them, and then grow them in 3D matrix using Matrigel to create tumor avatars for every patient. And we do that in high throughput and replicate so that we can create hundreds of thousands of copies of the same tumor to run high throughput drug screens.
So for modeling human disease, we take an organ on a chip approach, but it's a combination of organoids or organs on a chip. So when you're doing organs on a chip, you'll define a function of an organ. So something you can measure. So for the heart, it's contractility, so it's beating with force. That's something we can measure, and we'll focus on just making a tissue that has force in a petri dish and what we can measure of that.
At Cold-Spring we used patient-derived organoids that were grown in pure Matrigel in domes, and we would use those to predict cancer therapies for patients. The other approach that I've now followed up at the company that's called Xilis is they have developed something that is called a microorganosphere, which is an encapsulated cells in small Matrigel droplets so that you retain in a very small environment the cells that are coming from your biopsy or your resected tissue, and this can be now propagated. And this is kind of, I think, one of the key differences to many other companies and approaches that I've seen so far.
The models that we use are kind of twofold. We have normal lung organoids that we make, and we also make normal cerebral or brain organoids. And that's because we want to model the primary tumor. So for small cell lung cancer, it grows in the lung, right? And one of the main organs that it metastasizes to is the brain. So we like to have liver and brain and compare how these tumor cells grow within these different microenvironments and see how they have different responses to different treatments and therapeutic interventions.
We love patient and mouse organoid models. And so my colleague Josh Smith and I, who's a surgeon scientist and a colorectal surgeon, he's helped develop the Memorial Sloan Kettering Biobank for rectal tumor organoids. And we've developed matched tumor and normal organoids so we can assess relative radiosensitization on a patient-specific basis. We also have developed immunocompetent mouse models to study the interaction of radiation, also chemotherapy and targeted therapies with the immune system. So we can really kind of recapture and recapitulate the effect of what's happening not only in a tumor intrinsic manner, but a tumor extrinsic manner, capturing those immune effects.
So at Hub, I think most of the work consists of, on request of industry generally, to make specific models to ask specific disease questions. For instance, in inflammatory bowel disease, we need to study barrier function, so we make models in which we can actually measure barrier function, disruption or repair when we test the drugs. So making assays is probably one of the biggest things, and also where all the developments are by adding immune system, adding fibroblasts also, and to further develop these initial organoid models.
Corning Life Sciences is a leader in ECMs that are needed to grow organoids, such as the gold standard Matrigel matrix, collagen and laminins, as well as Corning Transwell permeable supports. In the scaffold-free space, we offer spheroid and alplasia platforms, clearing reagents for better imaging of 3D structures, organoid counting cell counter, and our newest launch, the Corning Matribot bioprinter. Therefore, our approach is to stay closely connected to our customers so we can provide these tools in a timely manner to enhance precision medicine.